أعوذ بالله السميع العليم من الشيطان الرجيم بسم الله الرحمن الرحيم السلام عليكم ورحمة الله وبركاته and hello everyone. So for today's lecture I will deliver a topic entitled non-imaging nuclear and molecular imaging, which one part of the subject under the course of PHMC 2172 PPPC 2721 nuclear pharmacy. So for today's topic, the things that we're going to discuss together will cover all of this. First, we're going to look into radioimmunoassay or RIA, and we're going to have a shilling test. And we're going to talk about how we use the nuclear method on measuring the blood volume and also the red blood survival. And then we're going to look what it's all about when we talk about the molecular imaging, and then look further into gene therapy and delivery. And last but not least, we're going to discuss about the nanocarriers imaging. So for these topics, we are not going to look into diagnostic in clinical and also therapeutic in clinical, which that particular topics will be covered by Dr. Zata Husna and Dr. Shahrir in their own respective lecture. So the first topic that we'll cover under non-imaging using a nuclear approach will be the RIA, or as I mentioned earlier, is the radioimmunoassay. So what radioimmunoassay or RIA is all about, let's look into the next slide. Okay, I'm not sure whether you guys are still remember, I did mention briefly about radioimmunoassay or RIA in the immunology course. Mainly for year 3, you have this in your deck, you just look back on it, but of course, it was really brief. I'm not sure about year 2 because your lecture has been combined both the microbes and also the immunology. So I'm not sure whether the one that you have it in the last semester covered about the RIA or even ELISA, but no worries, I'll explain in details about RIA so that you will understand what RIA is all about, what it's been used, and what the difference between RIA and other method. So basically, RIA it's actually a method to understand the antigen-antibody complex happen, especially you have an unknown sample, you don't know how much of antigen available in your sample, so you can use RIA as one of the methods. So if you haven't heard ELISA before, ELISA is also another method to understand whether how much antigen you have in your sample or how much antibody you have in your sample. But usually ELISA, either they use the colorimetric or they use the fluorescent excitation to give the value of the concentration. But for RIA, instead of using colorimetric or using the fluorescence, we are using the isotope to give you the values. And compare colorimetric, fluorescence, and radionuclide isotopes. Radionuclide give much sensitive value, meaning that if you have any slightly difference of concentration of antigen or antibody in your sample, it can be detected easily by radionuclide, so you have a much precise value and that's much more sense compared than ELISA that using colorimetric or the fluorescent excitation. Okay, I will not explain in terms of definition because most of this talk about definition, so mainly I will talk more details in the next slide, which you will refer to the schematic figure. I think it will be much easier for you to understand the method or the methodology of RIA in measuring or understanding the antigen and antibody complex. So let's go to the next slide. So as I mentioned earlier, like we are using this RIA to understand the unknown concentration of antigen or perhaps antibody in your sample, and then the value you get it based on the value that you got from the signal of the radionuclide or isotope that being used. So if you refer into this schematic figure, look to the left side here. So usually you have a antibody that you have the antigen that already been radio labeled here, and then you add the unlabeled antigen. So usually you unknown sample, you know how much antigen available in the sample, so you add them into a solution that have this labeled radioactive antigen, and then what happened here, they will start to compete. Okay, so the more unlabeled antigen you have in your sample, they will try to compete more, so then you will see they will replace of the labeled radioactive antigen on the antibody. So you have this blue color, the unlabeled antigen will compete with them. Then you have the free unlabeled antigen and also the free unlabeled radio antigen. So after the competition happened and then you allow the incubation, it depends. Sometimes they allow about half an hour, sometimes they allow about 1 hour, so it depends on the sample and the antibody you're working with, then they will precipitate this particular sample. So precipitate the antibody antigen complexes. So you have the what we call it the precipitation using the second antibody. So the second antibody will actually bound to this label antibody, and then this will be precipitate, and then this you will measure of this. So the unlabeled one you will available in the supernatant, so you have all the free antigen regardless it is label or unlabeled, and then one precipitation that you have this one that bound into the secondary antibody. So after this secondary antibody reacted with the first antibody, this one we will measure this one, we got the value whether how much of antigen available in that particular sample. So if you remember the one that we have a two sample, one is supernatant that has the free antigen, and then one you have the precipitate sample that you have the first antibody with the labeled unlabeled antigen, and also that actually precipitated by the secondary antibody. So the unreacted labeled antigen we call it as free antigen, which we denoted by F, and then the labeled antigen antibody complex or ABAG asterisk mark there we consider as bound antigen, which denoted by B. So when you have the B and the F, so the ratio of B over F give you the value of the concentration of unlabeled antigen in the mixture. Okay, so what this value is all about, it gives you the idea of the unknown concentration of antigen in a sample. And then usually how do you determine that? So you will make a standard curve. So I'm not sure whether you have a standard curve or dose response curve. This is quite common when you want to determine unknown sample, you have the known sample. So here what we do, you look into the graph on the left side here. So you have the standard curve that you prepared, and then based on standard curve you determine the value. So the standard curve, we use a known labeled antigen. Here we use labeled cytokine. So in this graph they use 1 to 5 iodine labeled cytokine and the unlabeled cytokine here. So they know the concentration. So here you can see the few plots based on that, and this give the value of it. So based on the standard curve, you compare this ratio that you have of B over F. That will give you the idea how much of the concentration of antigen available in your sample. So all the value that you got actually is from the signal from the radioisotope. So in this case, they use a 1 to 5 iodine. So it depends whatever the isotope you can use, but normally in RIA they use iodine 1 to 5 to determine the value of the unknown antigen of this. Okay, that's basically the RA is all about. So it's similar concept with ELISA, but then instead of depending of the colorimetric or the fluorescent excitation as their benchmark, we are using radioisotope as the benchmark to give the values, and then it's much sensitive compared than colorimetric and also the fluorescence excitation. So that's what we use. But of course, when we talk, if you look into details of the application, I'll explain after this. A lot of people have more options if they're using RIA or using ELISA. I'll explain about this in the next slide. Okay, so where exactly RIA being used as for application? Normally, we can use RIA not just understanding the antigen. We can use this particular method or particular assay to measure the hormones, concentration of hormones, concentration of enzyme, even steroids, and sometimes they also use RIA to measure concentration of peptides in a plasma. So because plasma have so many molecules available, and RIA will help on this to understand how much concentration available, and then how is it being used. It depends on the antibody being used. So they will use a specific antibody for particular hormones, specific antibody for particular enzyme or peptides, etc. And of course, we can use RIA to actually to determine how much of serum level of a drug that has been administered to a patient. So based on that, they also can understand after you administer drug to the patient and measure in the serum level of a drug in that particular patient. Okay, But there being quite a not a battle, but a being of discussion among the researchers or the clinician or even the scientists whether should we use RIA or ELISA. So it depends. If your facilities are not allowing to use any radionuclide, then ELISA is much safer. Again, because we are anti-radionuclide, you need to have all this particular safety. You need to have this the lead infused glasses, and also you need to make sure you have all the correct PPE to handle radionuclide. So if your facilities does not have or not equipped with this handling the radionuclide, then you can opt out using RIA. Then you can use ELISA as an alternative. So the main issue usually there using RIA or using ELISA is first we talk about safety, also we talk about sensitivity. Specificity is similar because we are using a similar antibody, but when we talk about safety, of course ELISA is much safe. It's safer because we just use the colorimetric and or the fluorescence material to give you the values, but with the RIA we are using radionuclide. When we talk about sensitivity, RIA is much sensitive compared than ELISA because of the slightly changes in the concentration will also alter the signal that you get from the radionuclide. So it depends on what you are looking for and then your application. So either ELISA or RIA, both of them can give a similar result. It's just whether safety and the sensitivity will be the main issues that you need to decide when you are choosing which assay you want to use for this particular application. So that's about RIA. Okay, let's go with the next assay still under non-imaging. So why I say it's non-imaging, because we are not looking into some kind of image produced by the signal of the radionuclide. We just want to take the value of or the value from the radionuclide. So that's why we consider as non-imaging. We are still using radionuclide, but then it's not coming into the photos or something. So the next non-imaging test that I'm going to explain is about the shilling test. Okay, so don't spell shilling the Malay shilling. This is the spell of shilling as CHILLILING. So that's shilling test. So let's see what shilling test is all about. So shilling test being used for a patient has particular disease, what we call pernicious anemia. So this particular patient usually has an issue of absorption of vitamin B12. So shilling test will determine whether this particular patient got a normal absorption of vitamin B12 or vice versa. So vitamin B12, another name, it can be called as cyanocobalamin. So you might find these people mentioned as vitamin B12, or some of them might mention the full name of this vitamin B12. So how exactly shilling test being conducted? First of all, you allow the patient to fast. Okay. Meanwhile, you have vitamin B12 that already being radio labeled with a tracer 57 cobalt. So what you do, you give oral administration to the fasting patient with this 57 cobalt labeled vitamin B12, and afterwards, after 24 hours, they will collect all the urinary excretion during the 24 hours, and then they will measure the signal or the value of the tracer. And this will be used as any test to indicate the status of the disease. And after 1 hour, you administered the patient with the radio-labeled vitamin B12. At the same time, you're going to intramuscular injection of unlabeled vitamin B12. So the reason why, again, we want to do some competition here, the labeled and unlabeled. So here, they will compete, the one that will be intramuscular and also the one you took it oral. And then afterwards, you look into 24 hours, they will take the excretion of the urine. And then for those who is considered has a normal absorption of vitamin B12, you will find the amount of the tracer in the urine sample is about a range of 10 to 40%, with the mean value at 18%. If you have issue of vitamin B12, okay, you injected intramuscular B12, right, the unlabeled one. So normally after through the oral, you go with the GIT, you will absorb about, and then the only left of the unabsorbed will be 12 around this range, 10 to 40%. But if you have abnormal absorption of vitamin B12, the value that we will found of the vitamin B12 inside of your urine sample will be much lower than this. So if you found it only about below than 7% of labeled trace of the tracer in the urine, so the patient can be suspected of pernicious anemia, and also it can be linked to other related disease. So a normal person, they will not absorb everything of the vitamin B12. So they have this range. So because here we have the competition of B12 that already bit intramuscular and also the one oral. But those that absorb too much, so this abnormality, we're talking about that you have too much absorption of vitamin B12. That's why we consider this particular patient is pernicious anemia, or perhaps other disease. So this Schilling test will tell a particular person whether they can be subjected to pernicious anemia or not. And of course, sometimes those already being confirmed pernicious anemia, they do this kind of Schilling test as well to observe whether there's some recovery or not. So whether this particular patient already been free from the particular disease or not. So you can use this method as well. So if I will show you the mechanism in the next slide in the schematic figure. So let's look at the next slide. So this is what I explained earlier. So you give a fasting patient with a radio-labeled vitamin B12, and then goes into the GIT and then the absorption and so on and so far. And then usually we expect the absorption happen at this area, the terminal ileum. At the same time, intramuscular injected of unlabeled being given as well. And then if you have normal absorption, you have 10 to 40% be excreted to urinary, but if you have abnormal absorption, you absorb almost everything of too much, then you have less of labeled B12 in urinary, meaning that you have some kind of issue of that particular disease. So that's a simple concept of understanding of the Schilling test. So the next non-imaging assay that we can use radioisotope or radionuclide as part of the benchmark of measurement is we can use this to measure the blood volume. So how exactly people determine someone's or a person have how much of blood. So you can use radionuclide approach to measure how much of blood available in a particular person in your body system, of course. When we talk about the blood volume, so in a normal human, okay, in a normal human, usually you will have about 4,000 to 5,500 milliliter of blood in your body, okay. But also this they give quite a range, but depend on the body size and also the age and so on. But this a normal range, so you have 4 liters up to 5.5 liters of blood in your body. And then why we need to determine blood volume, it's not like something that, okay, I'm free today, just want to see how much of blood volume I got. No, we not just use a easily way for you to measure blood volume without any particular reason. We use this particular method to measure blood volume, especially when we talk about someone who needs to go for the surgical theater. So they will need to determine how much blood volume needed pre and post-operative of patient care, because when you do a surgery, like they need to expect like if there is some loss of blood during the surgical process of the surgical procedure, so you need to have a quite amount of blood being prepared if those worst come to a situation happen. So sometimes you have quite a hemorrhage, so to ensure that you have enough blood being supplied based on the blood volume that they determine on this. And of course, it can be used for those who have a blood cancer, and then because of this method of measuring a blood volume, we want to understand how much of red blood cell available for those who have a blood cancer in the bone marrow, because usually if too much of red blood cells, it will causing your blood very thick, and it will affect the flow of the blood. And of course, we can also use this approach of using radioisotope or radionuclide to not just measure the volume of the blood, but also we can measure how much of red cell available in your blood. So a normal equation to understand how much of red cell volume available in your blood, you know the blood volume and you times of hematocrit, and then you times the factor of 0.92. That will give you the value of red cell available in your blood system. So the first method I want to introduce related with blood volume is using the 1 to 5 iodine serum albumin method to measure the blood volume. So the tracer they are using right now is 1 to 5 iodine. So how exactly we conduct this particular method? So you have the 1 to 5 iodine RISA. So actually RISA, the full name is radioiodinated serum albumin. So what you do, you administer it to a patient with 1 to 5 RISA, and then you allow that particular tracer to mix with your blood. So you allow it to recirculate throughout the blood. And afterwards, you will determine the blood volume from the radioactivity in the blood sample and the total administered activity. So that's a simple concept. So in details, how exactly you conduct this one. So you have how much of radioactivity that you need to have in 1 to 5 IRISA. Usually we have about 10 millicurie or 0.37 megabecquerel of 1 to 5 IRISA, and then you inject it or you administered to the patient intravenous, so by IV administration. And then afterwards, you're going to collect about 5 to 10 ml of blood sample in a tube containing heparin 10 to 20 minutes. So heparin to prevent from the blood to be clotted. So when you took the blood about 10-20 minutes after the injection. So you injected the RISA, and then after 10-20 minutes, you collect the blood sample of 5 to 10 ml. Make sure the tube got heparin. Okay, and of course, you're going to use the standard as the same amount as the injected dosage to measure. So usually, whatever it is, we're going to have our standard curve to compare with, so that you will know how much of the blood of the signal, and you determine from there. And then you're going to determine the hematocrit, and also the plasma will be separated. So how do you separate the plasma and serum in the blood? By centrifugation. And afterwards, you're going to take 1 ml of some plasma, and then 1 ml of standard, which you're going to count into sodium iodide selenium well counter. So this is how you're going to measure. So it's quite easy in terms of method of using this one, but the important part is like the preparation of the 1 to 5 iodine RISA of the radioactivity must around this range, about 10 millicurie. Then you're gonna have a proper signal afterwards. So this is the equation being used when you want to measure the plasma volume and then measure the blood volume. So you have first measure the plasma volume. You have CS. So CS is radioactivity in 1 ml of standard. VS is the volume of the standard corrected for fraction of injection dosage. And CP here is the radioactivity in 1 ml of plasma. So based on these numbers, you're going to have the amount of the plasma volume. And then after, to determine the blood volume, the value you got from plasma volume, you must divide it into 1 minus the total amount of hematocrit times 0.92. So what is 0.92? So this is actually a correction factor of how much plasma is trapped in the red blood cells, and also the differences between the venous and whole blood hematocrits. So this from this value, you will able to determine how much of blood volume in a particular person. Using the 51 chromium label red blood cells method, in terms of principle, it is similar when you want to use RISA. Okay, but the difference is in terms of radioactivity. I think for the RISA, you're going about 10 millicurie, but for this one, when you use the 51 chromium label red blood cells, you need to ensure that the radioactivity here about 50 to 100 millicurie, or you convert it into becquerel, it's about 1.85 to 3.7 megabecquerel. So about this amount of radioactivity, you're going to inject that intravenously into the patient. And of course, again, same with RISA, allow it to circulate, and afterwards, you're going to collect the blood sample. And how much, so when you should take the blood sample, about 15 to 20 minutes after the injection, and then you're going to determine the hematocrit. Again, you're going to have a standard. We need to split this the blood to have the plasma amount, the plasma sample, the whole blood sample, and the standard. So again, you're going to measure using sodium iodide selenium well counter. And then from there, you're going to measure the how much of blood volume available. In terms of the formula or the equation, it used a same formula or equation that you use for the result. So what the difference between this method and result method is just what you are using as a tracer. The one that we have in the previous one, we used the radioiodinated serum albumin. For this one, we have the red blood cell being labeled, and of course, that one being labeled with a tracer of iodine or iodine. This one being labeled with the 51 chromium. So that's different. Concept, principle, equation, similar. Interestingly, a same kind of method of radio labeled of non-imaging can be used to determine of the red blood cell survival of the red blood cell age. So you can actually determine of the age of your red blood cell using radionuclide. So it's not just we're talking about amount of red blood cells, we talk about the blood volume, but we also can determine the age of our RBCs. So in general, our red blood cell in terms of lifetime is about 120 days, which is about 4 months. So, but also there's some slight difference subject to individual, but this in general. And then we also know that even though we said the lifetime of red blood cell RBC is about 4 months or 120 days, we cannot say that they are all every time, every 4 months they are freshly made and 100% they are fresh or same batch of red blood cells. So meaning that in our blood, you're going to have a variety of different ages of our red blood cells, and then we can measure of the average survival of half-time of these red blood cells by using the 51 chromium label red blood cells. So how exactly you conduct this? Again, you have red blood cells, you label them with 51 chromium. In terms of the radioactivity is about 50 millicurie. Then you inject into the patient intravenously. And then you're going to collect the blood sample after 24 hours. And after 24 hours blood sample, you consider that as 100% of sample. And afterwards, you're going to take about 6 to 10 ml sample every 48 hours until the last sample that I took have half of the activity of the 100% sample. Meaning that, for example, after 24 hours, you got 20 millicurie, an example, okay? And then you need to ensure that where you want to stop into taking the blood samples every 40 hours when it reach 10 millicurie and below. So then it gives you a signal that, okay, that's enough. That will be my last sample of the blood. Now, of course, you're going to hemolyze your red blood, and then you're going to do a well counter to check at the activity. Okay, compared with the blood volume, because the period of conducting the assay about a same day, and then you took the sample after 10 to 20 minutes. But this one, because you're going to have 24 hours and every 48 hours, every sample that you took, for example, the 100% 24 hours, immediately you're going to count the radioactivity. And the next 48 hours, you got the sample, immediately going to count. The reason why we want to avoid any decay correction, because you know that radioactivity have their own half-life, and then you want to avoid that radioactivity is lower, not because of the effect of the age and so on, but actually it's the effect of the decay. So that's why every sample that you're going to take for this one must be counted on the same day, or I should say impromptu or immediately. After you end taking the blood sample, you took all the values, you're going to plot then on the graph. Okay, plot all the activities on the graph, and then how you plot it, you look into the left graph here. So you have time of days, also the rates, the percentage of red cell survival. So this one usually, if you consider as normal, so our normal values range of our half-time for red blood cells usually between 25 to 33 days with a mean value of 28 days. But if you are considered as hemolytic anemia patient, your half time will be much shorter, and then you're going to have something much lower. So that's why we consider normal, because like half mean about 50% around these days, so 50% around 28 days. But then if you have your 50% below than that particular days, for example, here the abnormal one, you have about that at below than 16 days, you are considered as a hemolytic anemia patient. So you will be subjected to this particular disease. So that's how exactly by using the radio-labeled red blood cell can help you to understand your age or your survival of the red blood cells, and of course whether you are considered as normal or you'll be subjected to this particular hemolytic anemia disease. Okay, that's end of the first part of the non-imaging approach. Now I'm going to focus on the second and third part, which are nuclear and molecular imaging, where exactly how exactly from the radioactivity from their signal can be visualized. So that's what we are going to look all about on this particular part. In general, molecular imaging, or I use as MI here, is one of the methods that we use to determine the spatial or temporal status of particular molecular or even the cellular processes in the normal or abnormal state in living organs. So from the radio signal that we have, perhaps that goes to particular tissues or cells in our body system, from the signal we change into our develop into a visual where you can identify mainly in target molecule in specific organ or target molecule in specific disease, or even you can use this molecular imaging to develop a high infinity probe for the molecule. And of course we can also understand particular distribution and also pharmacodynamics of molecules that are being administered to a patient. So that's how the MRI can be used in general. There are a few types of techniques available for the MI. Some of them that are listed here is what we call it magnetic resonance imaging, or MRI. You also have optical imaging. We're going to have also ultrasound, and we also have Raman spectroscopy, and another one what we call it nuclear medicine. So in nuclear medicine, there are a few things that we'll explain after this, which in details it will be explained by Dr. Shahril under his lecture about the diagnostic in clinical application. Briefly, when we talk about of MI in nuclear medicine, there are a few things that you need to understand and need to be focused on with. First of all, when we talk about MI in nuclear medicine, they will use a high affinity probe, which a specific target to particular tissues or cells. And of course, the probe that we use must have the ability to cross the barriers. For example, it can cross the vascular, it can cross interstitial, it can cross cell membrane, so that that particular molecule that we use as tracer can bound to the particular area or target, and perhaps some kind of cells or tissues, and then it will localize that. And of course, when we talk about localization, the probe must be localized at a maximum amount that they're able to do so. So for example, like you have a particular drug A with a tracer that only bound to the EGFR expressed breast cancer. So when you administer to a patient through IV, that particular probe will only localize at EGFR express breast tissues, breast cancer tissues. So then you will able to localize where is the location of the tumor of the cancer, and also the size and how the condition of it. And of course, last but not least, the image technique must be sensitive to actually measure the probe that being used into or administered into the patient. So there are a few techniques that being used in nuclear medicine when we talk about MI here. So we got SPECT-CT, we got PET-CT, we got PET-MRI. So what are these SPECT-CT, PET-CT, or PET-MRI? I will not talk in details. This, inshallah, will be explained by Dr. Shahril. What's the differences? SPECT-CT or SPECT only, SPECT-CT or PET only, or CT scan only, or MRI only, or this combination. So you will looking forward for this particular lecture with Dr. Shahril, because he's our nuclear medicine, our own hospital. So you can ask a lot of questions from him related to this. As the techniques of the MI imaging will be explained by Dr. Shahril, on my part, I will focus on gene therapy and also gene delivery, which related to precision medicine. I'm not sure whether you guys have heard this term before. So precision medicine is some kind of like a customized medicinal approach where you can custom each treatment approach for each particular patient. Rather than use particular drug A general dosage to everyone, you can have a proper customization of therapy for particular patient. So meaning that you will avoid less inefficacy, you can avoid in terms of costing of using drugs and so on. So that's what a lot of clinicians, scientists, and researchers try to work on it. And one of it of this precision medicine is delivering gene as a gene therapy to provide the next future precision medicine. So when we talk about gene therapy, the first of all, we need to understand on certain diseases, they are actually caused by defects in genetic constructs. Especially when we focusing into of genetic deficiency, the aim of gene therapy actually to restore back the function that caused by genetic mutation. So that's how exactly gene therapy comes out to help on this. But if you focus in more details in oncology or in cancer, we understand that there are certain genes that are responsible causing particular cancer, and certain genes can also responsible to actually modify the cancer or the tumor cells. So we already found out that certain gene can prevent growth of tumor cancer, and some of them can also promote of the host cell to generate anti-tumor activity, meaning that by you generate certain genes, you promote the certain areas of the host cell or tissues to generate anti-tumor activity, so meaning that it will do all the killing of the cell cancer without using any drugs. And also with gene therapy, on top of in cancer, we can use this to actually delay or even inhibit certain progression of chronic disease that caused by specific genes. So meaning that by delivering certain genes, you can actually help people in cancer mainly, and then for those who have been suffering by the disease caused by specific mutation of genes, we can use this approach of gene therapy. If we talk in detail, so gene therapy has two approaches on delivering them. So either through ex vivo or through in vivo. So what is ex vivo? Ex vivo, usually you have particular target cells or tissues, you remove them from host, and then you transfect that particular cells or tissue with specific gene, and then you redeliver them back again to the host, and they will do the work. For example, you take some immune cells outside, and then you transfect them with particular gene, perhaps to promote interval anti-tumor signal, and then when you got this particular signal being developed with the immune cells, and then the immune cell will be reinjected back into the host, and then because they already got signal of anti-tumor, they will start doing the killing of any cancer cells available in the body system. So that's ex vivo. So when we talk about in vivo, so you're going to deliver the particular gene using a particular carrier to appropriate target cells. It can be through oral administration, through intravenous administration, intratumoral or intramuscular. It depends on your target cells or tissues. And this particular carrier, so I want to focus on this part mainly. This particular carrier, we call it as vectors. So what you do, you have a gene of interest that you want to deliver. You package them into a particular vector, and then you're going to deliver them to the target cells or tissues. And this particular vector can be either viral or non-viral, and that's how I say it can be delivered via inhalation, intramuscular, intravenous, or even intratumoral. It depends on the target cells and tissues. Okay, let's talk into details about this particular carrier. First, we look into the viral vectors. There are a few viral vectors that are being used to deliver gene. First of all, I will explain is about adenoviral. So adenoviral, I hope you guys remember back whatever you learned back in microbe subject. So adenoviral is a type of virus that has double-strand DNA, but they are really integrated into the host DNA. However, they can elicit strong immune response, which actually worsened when it's been administered repeatedly. So what you do here, so the adenoviral or the virus, adenoviral virus, you remove the viral gene, which required for replication, And then what you do when you remove that particular gene, you're going to replace that gene with your interest gene of interest into them. So instead of when they go into our body and that they deliver the genes that cause the disease, they're going to deliver our genes of interest, for example, for curing, for cell killing, and so on, and then it will do the work. So that's how we use the adenovirus. And the size of the adenovirus is quite big because they contain double-strand DNA, meaning that it allows much more capacity to encapsulate or to insert a lot of genes inside of it. And this particular adenoviral of this virus can be produced in vitro culture media. So meaning that you can easily make into the lab and then can be used for further application, preclinical or even clinical application. So the second viral vectors I'm going to introduce here, what we call this as retroviral. So retroviral compared to adenoviral, which contain double-strand DNA. Retroviral only contain a single-strand RNA genome. So when it transfers into the host tissues or cells, it will convert it into DNA, and then we incorporate it into the host DNA. And then this particular virus are replication deficient, and this also retroviral it also been used quietly in both experimental or clinical and also clinical. Usually the retroviral genes, you will find it the name by GAG, POL, and ENV. And this particular gene, you're going to replace with your specific interest of gene to be delivered by the virus to the host cell. And they also can be easily be made into the laboratory. But however, this particular retroviral, because it's only contains single-strand RNA genome originally, so meaning that the capacity for you to insert other genes inside are very small. So meaning that it can be limited only by small size of gene that can be inserted, and any large genes you can't use this retroviral as a method of delivery. So meaning that you need to use adenoviral instead to deliver large genes into the host cells or tissues. Another additional point I would like to add for retroviral, so compare with adenoviral, they can infect as much as possible into the whole cell or tissues. But with retroviral, they can only infect of the dividing cells. So if the cells are not dividing, they will not infect. So for example, in the laboratory, we use a retroviral vectors called as lentiviruses. So lentiviruses is commonly being used in laboratory to transfect the cells. For example, you have particular cells. What I experienced have what we call the breast cancer MDA231. MDA MB231, so this one particular breast cancer, then we want this particular cancer to express certain receptors. So what we do, we're going to use lentivirus, deliver the particular gene that responsible to deliver to express the particular receptors. Then we use the lentivirus, carry this particular gene, transfer with the MDA MB231 cells, and then afterwards we have MDA MB231E, which is going to express EGFR. So that's how we do. And also another popular lentivirus that commonly used is HIV. So HIV also, if you modify them, remove all the viral genome, and then you can be used them to deliver intended gene. And interestingly, HIV, one of the unique retroviral that can both transfect or infect dividing and non-dividing cell, and will not induce any immune response. So that's how exactly when you got infected by HIV, it goes everywhere, and then it will not trigger any immune response, but it actually damages your immune response. That's why we call it immune deficiency virus. So this the schematic figure of these two particular viral vectors. You have the retroviral vectors RNA genome and the noviral vectors DNA genome. So usually with the RNA genome, you have an envelope here and then RNA genome inside. Also, you have envelope protein outside too. But with the DNA genome or the vector, you don't have envelope, but instead you only have the capsid protein that protect the DNA inside. I think this really refresh a bit for you if you remember your microbe course. I think that year 2 should remember more than year 3 because you just took it last semester, right? So I hope this kind of helped you to understand further about this. So this is another refresh how exactly the transfection happens. So this is for the retroviral, so you can look into the schematic figure. And this one is the schematic figure of how is the adenoviral being used as a carrier to deliver genes and how it transfects into the cells here. And here is a table of different non-viral vector that you use to deliver genes. So I'll explain some of the example of non-viral vector in the few slides after this, but this is a bit of summary that you will understand how exactly you use different non-viral vectors to deliver genes to particular diseases or particular tissues, even what they are been delivering, what are the advantages and so on. And even they explain here in terms of key mechanism, how exactly they've been delivered. And now I will talk about details of these non-viral vectors. So we are using these non-viral vectors as nanocarriers for imaging. Okay, where you're gonna merge the knowledge of nanotechnology in medicine, which actually that's my research expertise. For those who might interested to do research with me in future, please do come into my group, which we are really focusing into nanomedicine and drug delivery, which include we are using radioisotope and radionuclide as part of the research studies. What is nanocarriers, nanoparticles is all about? So nanocarriers or nanoparticles is mainly the particles of a structure that have one dimension of size 100 nanometers or less, but you might find some up to 300 nanometers, and then it can be made from variety of materials. You can have polymer base, you can have dendrimer base, you can have lipid base, for example solid lipid nanoparticles in the liposome. You have nanocarriers that are made from inorganic materials, including carbon nanotubes, magnetic nanoparticles, or even you can have nanocarriers that are from biological materials as virus-like particles. So this not considered as viral vectors because it's mimicking the virus but not a virus. And also we're going to have nanocarriers that are made from protein. For example, you can have something made from albumin to make into a nanocarriers or nanoparticles. This is a summary of what I mentioned earlier. So you have the biodegradable polymeric nanoparticle here, you have dendrimer base, you have solid lipid nanoparticle, you have made from liposome, and you have magnetic nanoparticles here. You have the carbon nanotube, and that's why I said virus-like particle and protease nanoparticle. So during my PhD, I've been exposed with many types of nanocarriers, but mainly my work related with this virus-like particle. But now in my group, research group, I will explain after this at the end. Uh, we are mainly focusing into the biodegradable polymer nanoparticle as a combination with other nanocarriers, as we are developing hybrid nanocarriers for cancer mainly. What are the advantages of using nanocarriers especially for imaging? We talk about here from the point of view of nuclear. So because it got a small dimensions in terms of size, as I mentioned, it's about 1 to 100 nanometers, bigger about up to 200 nanometers. Got this small size, meaning that it allows you to penetrate into tissues and cells easily, meaning that anything that you delivered can be penetrated easily into certain tissues and cells. And of course, because like based on modification that you've done with non-carriers, you can alter drugs pharmacokinetic. For example, you have hydrophobic drugs. So hydrophobic is hard for you to administer through IV, and then it means that it's hard for the drugs to be delivered to targeted cells and tissues. So by using non-carriers, you can help deliver the hydrophobic drugs, and then instead of a got problem into our bloodstream, easily it can go to bloodstream and only arrive at specific target or cells. And this one I mentioned earlier, it can be for various molecular structures. So how you choose the type of material to develop these nanocarriers, it depends on your application and also your target cells and tissues. And of course, because it's kind of like a very tiny ball, you can not just delivering drugs, you can deliver other biomarkers like protein, DNA, gene, even plasmid, even you can deliver any other imaging probes carriers, including radioisotope. So that's kind of advantage that you have. Then a lot of possibilities for modification, meaning that it's really will be helpful for the further imaging, especially when we talk about cancers and so on. I'm giving here some examples of nanocarriers that have been used. So first I will introduce what we call polymeric nanoparticles or polymeric nanocarriers as part of the non-viral vectors. Okay, so usually you're going to find these polymeric nanoparticles or polymeric nanocarriers, or sometimes they call nanocapsules, in terms of size about 10 to 1000 nanometers. So you can have the drugs dissolve, entrapped or encapsulated, or even attached to the nanoparticle matrices. So if you make it a nanocapsule, you have empty space inside. The core can be encapsulated with the drugs, or you're going to have upon nanoparticles have a matrix then going to mingle around into the drugs into the matrices and so on. And because of small size, this particular nanocarriers has a huge surface areas, meaning that you can also allow the drugs or any particular molecular can be absorbed on their surface. And for these particular polymeric nanoparticles or nanocarriers, it can deliver drugs, proteins, and even DNA to target cells and even organs. And because it's from polymer base, it can also promote effective permeation to cell membranes and stability in our bloodstream, especially when we talk about delivering a hydrophobic compound or drugs to the body system. These are some example of polymeric nanoparticles and carriers. So you have a version of nanosphere. So this is what we call the matrices, polymeric matrices, and then you have the drugs mingling around the matrices. Or another one that I mentioned, also the nanocapsule. So you have the outer layer. We have the polymeric membrane, and if you have the inner core, then you can put the drug inside. Actually, this method really helpful if you want to talk about easily digested drug or hydrophobic drug, which you're going to find a lot of difficulties when you want to only deliver the prodrugs into our body system. So as I mentioned earlier, nanocarriers can deliver imaging probes, including radionuclide radioisotope. So here, examples of polymeric nanoparticles in nuclear medicine, we have 99 technetium labeled PLGA nanoparticles. We can also find 111 indium PLGA packed nanocapsules. So what they do on this one, they use for diagnostic, also for therapy as well. So they become the tracer. So they're going to trace where this PLGA nanoparticles or PLGA packed nanocapsule we're going through into our body system, whether it's going to be localized in specific tissues and organ. So here an example, you have a polymeric nanoconstruct which you loaded with anti-cancer drug, we call it docetaxel, and then is DTS, and also curcumin for cancer combinatorial therapy and nuclear imaging. So the polymer itself delivers this drug for therapy because you labeled them with radioisotope. You can image them, localize or track where this nanocapsule have been went through, whether it's localized in particle cancer or not. So we have another example here. We have PLZ nanoparticles with biotinylated F18. This one is used to deliver to the brain for PET imaging. So they use this particular F18 to deliver these nanocarriers to the brain. And another example here for MR imaging, we have PLGA packed nanocapsule that being labeled with 111 indium here, and also they got a magnetic nanoparticle inside of it. Another example we use for SPECT imaging, you have PEG polymer nanocapsules, specifically targeted with folic acid, and then they radio labeled them with a tracer, and then they image them under SPECT imager. The next nanocarriers I'm going to introduce is the liposomes, or it's a lipid-based nanocarriers. So liposome, they are made from a bilayer of phospholipid, which surrounding a central aqueous phase. So with the liposomes, usually the center of this particular carriers is aqueous phase. So you can create this liposome from cholesterol and even other natural non-toxic phospholipids, and because of their size and they're also hydrophobic and have also hydrophilic character, uh, liposome is really a promising system for drug delivery because of the bilayer. You have the hydrophobic and hydrophilic. So the hydrophilic will be the outer layer and the hydrophobic inside of this liposome. And then you can change the composition of the liposome based on how much of lipid you want to use. You can choose surface charge, even the size, and based on the method of preparation. And because of liposome is a lipid layer, so it's more fluid, and then it's kind of flexible. And then you can also change the charge of the bilayer. So if you look into the photos on the left side here, these are example of the liposome. So you have the empty liposome here. So the pink one is the phosphate lipid layer, and the green one is the aqueous phase. And then this one you have the magnetic nanoparticles inside of the liposome here, which is the blue color. So here it shows that the flexibility, you can see even the shape here is empty, the size can be quite fluid, and then if you have particular something inside, it's much more rigid. Examples of liposome that being used in nuclear medicine, we have a 111 indium liposome, we have 67 gallium liposome. Also, we also have the 99M technetium liposomes. So again, similar with the polymeric and nanoparticles, nanocarriers, liposomes labeled nanocarriers also can be used for diagnostic and therapy. For example, here you have an immunoliposome that displayed HER2 antibody for breast cancer therapy. Also, liposome that encapsulated with doxorubicin or DOX for which is anti-cancer drug for tumor killing. You also have a liposome that display anti-EGFR ligand, meaning that you're going to specific bound to overexpress EGFR cancer cells. Also have 111 indium liposome that we use to track localization of A549 is a lung tumor in vivo. So these are examples of using this liposome, which the trees are here. So you have all of these delivered drug that also radio labeled with this either 111 indium, 67 gallium, or even 99M technetium. And then you can image them. For example, I have experience using the indium and also the technetium. You can image them under SPECT-CT imager, which give you the localization of the particular cancer in the animal. This is an example how they developed the liposome. So they use the SPC and the SPPAC 2000 melamide as their the phospholipid layer, and then they have a particular compound A20FMDV2, which they actually conjugated on the surface of the particular liposome. So they conjugate at this melamide peptide here. So the example of how they develop one of liposome, and then you can have other tracer to put it together. So you're going to have a tracer that are going to radio labeled with this liposome. So that's marked the end of our lecture for today. So I mentioned earlier, I have my own research group. We call it INAREC, Izzat Nanomedicine Research Group. For those interested, perhaps in the next semester, want to do LS and RP with me, feel free to come and visit the research group. I'll be welcome all of you to join the group, even in year 3, perhaps you're going to do RP or LS with me, or perhaps you are thinking of pursuing your study in master and research and want to do further understanding of nanomedicine. Feel free to come to and join my group. At present, we have nearly 13 to 16 people in the group, range from the LS, RP, FYP from other Kulliyyah, and also postgraduate student from IIUM. Also have students from UTM too. So I think that's it from me. If you have any questions, you can use our Telegram group available in the Telegram. You can ask question, or you can direct PM to me, or you can send me an email here. This is my email, or you can contact me through this 4 social media. I have Facebook, I have Instagram, I have Twitter, I have a LinkedIn. So feel free to use any method available for you to ask me questions. And because now we couldn't have a face-to-face lecture, I'm gonna have for this course all asynchronous and recorded lectures. So if you don't understand, please don't wait too late. Just ask me, and I'll be welcome for you to ask any questions. So that's it from me again, and I wish you all the best, and shall see you in the next lecture. Assalamualaikum warahmatullah, and have a good day.